So I'm going to give the research talk, a sort of an introduction to the ideas, and then maybe after dinner, depending on what people want, I can get into more of the technical details of the statistical analysis, which are quite important with these next generation sequencing technologies. Okay, so I just will start with an introduction to what we do, all right? Broadly speaking, oh my God. Broadly speaking, what we do is we dissect mechanisms of gene regulation, so transcriptional, co-transcriptional, post-transcriptional, and also try to understand the dysregulation of gene expression in cancer, all right? So we also, one thing that's special about what we do is we use machine learning tools. So when we try to understand, so it doesn't work? Okay. Other way? Huh. Okay. So we try to understand mechanism, and in particular the sequence information that mediates regulation at different steps by posing supervised learning problems. Okay, so you could imagine that, as you maybe discussed a bit today, if you have a promoter and you have some kind of expression output from the promoter, okay, you could ask, can I determine what sequence elements exist in the promoter and maybe other methylation state that will predict the transcriptional output, genome-wide? Or if I look at the 3' untranslated regions of transcripts, can I figure out what are the sequence elements or contextual features of predicted miRNA binding sites that will help me predict the extent of downregulation, right, mediated by miRNAs. So we can set up prediction problems and infer some kind of mechanistic information by looking at the models. All right, so I'm not, this next hour is not so much about that. But let me just tell you a little bit, a little overview of some of these different topics before we zoom in on one topic, which is this co- and post-transcriptional regulation. Okay, so we are actually very interested in the context of ENCODE and our other work in understanding cell type specificity of transcriptional programs. So to do this, we're looking a lot at the dynamics of DNA accessibility using DNA-seq data. So you can imagine in, say, differentiation in the hematopoietic system, if you see where the DNA's hypersensitive sites are present, where they're first established, where they're turned on and off. Okay, so we get some idea of what, when are transcription factors binding and holding these regions of chromatin open. Okay, and what we're interested in doing is understanding the dynamics of these DNA accessible regions. They are a way of mapping enhancers and promoters, and understanding the sequence information, the DNA sequence information in terms of transcription factor binding. Okay, that sort of orchestrates differentiation. Okay, and as a proof of principle last year, we looked at DNase and a sort of late differentiation step in the T cell lineage. So we were looking at CD4 positive T cells versus regulatory T cells, and we were showing that some DNase regions, even ones that are occupied by the master regulator of this particular cell type, regulatory T cells, are already accessible in CD4 positive T cells. Okay, so there's poising early establishment of regulatory regions, and potentially a situation where at in different cell types, different factors are holding these regions open. Okay, so some things have to be set up earlier in differentiation. All right, so I'm not going to talk more about that work. We're also interested in cancer systems biology. There's still an option that we could talk about this after dinner. Again, trying to understand tumor versus normal expression changes, both in terms of information at the promoter, transcription factor binding sites, and information in the 3' untranslated regions, microRNA binding site. Okay, and these models use structured sparse regression. Okay, so this is definitely a machine learning approach. So we can talk about that maybe if we have time. Okay. All right. But what I'm going to talk about today is actually just new technologies and looking at new questions that are enabled by next generation sequencing protocols. Okay, and I'm going to talk about two projects. One is published, one I hope will be published soon. Okay, I think we're close. The first is called ClipSeq technology for mapping the binding sites of RNA binding proteins transcriptome-wide. And the second is 3'-seq for mapping the 3' ends, and in particular the alternative 3' ends of transcripts transcriptome-wide. Okay, so only a bit of machine learning for the next hour and more about what we can learn from these technologies. Okay, so last few slides of introduction. Okay, so I call this talk my regulatory transcriptomics talk, but that's just a term I made up, all right? So what people talk about usually is regulatory genomics, all right? And if you look at most of these really big projects, like ENCODE, a lot of next generation sequencing technology goes to mapping the epigenetic state of cells, all right? So the genome-wide mapping of the occupancy of specific transcription factors, or histone ChIP-seq for mapping histone marks, DNA-seq for, again, the DNA successible regions that are promoters and enhancers, MNA-seq for nucleosome positions, all right? And these are really mature technologies. They work well. They're huge public data sets that you can exploit. And they're all about really the study of transcriptional regulation, cause and effect of transcriptional regulation. All right, there's still more to come. I mean, I think the next big thing that is unexplored territory, and the technologies are not very mature, are for capturing the 3D structure of the genome, chromatin conformation technologies, so high-Z, chia-PET, 4C, 5C. Okay, so there's still more coming in this endeavor. But what I'm going to talk about today is that there are many layers of regulation of gene expression after initiation of transcription, right? So often we think of post-transcriptional regulation. So after the transcript is already made, there's regulation of export, it's localization to different cellular compartments, RNA degradation, initiation of translation. Many of these processes are mediated by the binding of RNA binding proteins, often via signals in the 3' untranslated region. Okay, and the most famous example is microRNA-mediated gene silencing. Okay, so as you probably know, microRNAs are loaded into a silencing complex called RISC. One of the proteins is argonaute, okay, and it leads to both degradation of the transcript and also inhibition of translation. I think that's fair to say. Right? So we will be talking about microRNA-mediated regulation, but there's also co-transcriptional regulation. Okay, so I think 10 years ago we didn't have this picture of co-transcriptional regulation. Now we do. That many processes, including alternative splicing and alternative cleavage and polyadenylation. Okay, so where you cleave the transcript, add the poly A tail to define the 3' end of the transcript. All of this is happening by machinery that is kind of partially recruited by the polymerase and dragged along with it. All right, so it's all happening. You're elongating the transcript, and this machinery is making choices about splicing and about recognizing polyadenylation signals and cleaving, okay, all at once. And we're actually going to talk about APA, this process of choosing the 3' end. Okay, and it's regulatory transcriptomics because we have new transcriptome-wide technologies to map these regulatory events transcriptome-wide. So these are and post-transcriptional regulatory events. Okay, so I'm going to talk about ClipSeq, and in particular, differential ago ClipSeq to define a full set of miRNA target sites biochemically for 1 miRNA in a given cellular context. And I'm also going to talk about 3'-seq. This is a new technology for mapping the 3' ends of transcripts and allowing us to see tissue-specific and stimulus-specific alternative cleavage and polyadenylation. Okay. All right. So defining miRNA's transcriptome-wide binding sites with ClipSeq. Okay, I think everyone knows what miRNAs are. Right? If you don't, you know, miRNAs are a class of small regulatory RNAs. In their mature form, they're 21 to 23 nucleotides long, loaded into RISC. They have a different, they're transcribed, there are miRNA genes, but they have a different biogenesis pathway than other coding transcripts, right? And they are implicated in many processes. So they're involved in regulation and development, cell differentiation, regulation of cell cycle and apoptosis, initiation and progression of cancer. Some miRNAs identified in progression to metastasis. Okay, so miRNAs post-transcriptionally downregulate their targets. Okay, so this is the paper that I read when we started doing work in miRNAs some time ago. Okay, so this is a landmark paper of Lee Lim in 2005, and he was studying the genome-wide effects of miRNAs for the first time. Okay, so what he did was he took HeLa cells and he transfected pretty high concentration a single miRNA that wouldn't normally be in the cell. Okay, like miR124, we think of it as a neural specific miRNA. And then what he saw, right, is that if you wait 24 hours and you measure mRNA expression changes, you see hundreds of targets mildly downregulated. Okay? And if you take the 3' untranslated regions of these transcripts, and you do that, these ones that go down, okay, and you do a simple motif search approach like MEME, what you see is you find a signal, you find enrichment for a motif that is complementary to positions 2 through 7 or 2 through 8 of the miRNA. So this is called the seed region, this is called the seed match, okay? And this was known to be the primary determinant of miRNA specificity. Okay, so at the time, this was news to the miRNA community that you could see effects at the mRNA level. These seem to be direct effects because we're seeing the seed enrichment. You have hundreds of targets, not dozens. Okay, so this was the first, genomics meets miRNAs. All right, but the thing is that the seed pattern is neither necessary nor sufficient for miRNA-mediated regulation. right? So if all you had was this seed pattern, either the 6-mer or 7-mer, you'd find it everywhere and predict thousands of targets, and you'd be wrong most of the time, right? Conversely, you can't assume that every target has a seed, even a 6-mer seed, because through the literature and through experiments, we know there are non-canonical seed patterns. So this is sort of a mystery. What is the full set of rules that govern microRNA-mediated regulation? Though most computational methods will just use conservation and a seed dressed up with a little, you know, maybe a few other contextual features. Okay, so we started on this project. This is a collaboration with Sasha Rudensky's lab. He was interested in miR-155 in the immune system, in particular in activated T cells. So in T cells, but also many other immune cells, macrophages, dendritic cells, B cells, when you activate them, they ramp up the expression of miR-155, all right? So upregulated upon activation of many immune cells. They're also, miR-155 is also overexpressed in many cancers, and higher expression promotes autoimmune inflammation. Okay, and then there's this very nice phenotype, so you can make a miR-155 knockout mouse. It's viable, it looks okay, but you present an immune challenge like salmonella, and the knockout mice all die. All right? So while wild-type mice will survive. So, so important. Okay? So what we wanted to know was, what are the targets of miR-155? What are the full complement of targets in activated CD4 positive T cells? Okay, so we used a new technology called ClipSeq. There are different versions of CLIP. This stands for cross-linking IP followed by sequencing. We use a variant of the HITS-CLIP approach that was developed by Bob Darnell at Rockefeller. There's also PAR-CLIP, developed by Tom Tuschl's lab. There's another variant of HITS-CLIP called ClipSeq by Jean Yo. Okay, so, and actually there are only a few labs in the country who have this working. It's not so easy. All right. So what you do, you take your cells, you cross-link the RNA binding proteins to the transcripts with UV. You do an RNase digest, and you have pull down, you IP for Argonaut. Okay, and then okay, so this is important. You get both the microRNAs and the mRNAs that are pulled down with this complex. But then you separate them out in a gel, and you sequence the microRNAs, and you sequence the mRNA, the fragments that were sitting under Argonaut, but you lose the association between them. All right? So we'll sequence the microRNAs that were loaded into Argonaut. We'll sequence the reads that tell us where Argonaut was sitting, but we no longer know what microRNA was sitting in Argonaut at what position. Yeah? Okay? There is a new protocol that introduces a ligation step, so you ligate the microRNA to the target sequence, and you get chimeric reads, so you can see some of these associations, but in the published data, the ligation efficiency is 2%. Okay, so you see some, but not everything. Yeah? All right. So what's special about our paper is that we actually did this in 2 cell types. We did differential ClipSeq. So we did in wild-type cells and also miR-155 knockout cells, so activated T cells from these wild-type mice versus the knockout mice. Okay, and what I'm showing you here is just a little piece of the UTR of some gene, right? And I'm stacking up replicates. And this is the signal we're looking for. We're looking for a nice binding peak. AGO was sitting there when 155 was in the cells, but in the cells that don't have 155, we see significantly less read evidence of binding. Okay, so this is a differential peak We call it a miR-155 dependent peak. Okay. All right, so I just am going to tell you a little bit about how we do this analysis, but just really a summary, and tell you what we find. And then, depending on what we do afterwards, I'll give you some details about how you do this. This is a case where the statistics matter. If you, say, assume your read counts are log normal, and they're not, you actually won't find, you won't get the answer, okay? All right, so some of the basic analysis challenges. So this is, I guess this is HIT1 alpha. Okay, so this is what the data actually looks like. So you see a lot of AGO binding, right? So here's a nice differential peak, right? But in general, you have very complex peak profiles. And I think a lot of RNA binding proteins look like this if you have enough coverage. Okay, so actually, none of the existing peak calling solutions, so you've probably seen in ChIP-seq, there's MAX, there's SPP, there are various methods for finding peaks, but they don't work, or either because they're using things like cross-strand correlation that we don't have here for RNA binding, or just the assumptions are wrong. So we actually do an edge detection routine. We're finding these regions where there's an edge in the signal. So like it's low and then very high. Okay, this is from computer vision. And it works fairly well. So we can kind of split up peaks. We can, if we see one peak right beside a lower peak, we can detect those multiple edges and break them up into different AGO binding footprints.
So what's the scale here? Is this an higher UTR?
Okay, so I'm showing you a piece of the UTR, and I think I've cut off the scale. Yeah, but the peaks are 30 to 50 nucleotides. Okay.
Those two vertical lines.
This is, yeah. So this might be 30, yeah. Something like that. This is the actual 7 mer or 6 mer seed, okay? But as we'll see, we have peaks like this where we see binding in the wild type and not in the knockout that don't have seeds, okay? All right, so we have to define these events, right, that are AGO, footprints of Argonaut binding, and then we have wild-type replicates. We actually have 12. I'll tell you why we have so many in a second. And 12 knockout, all right? So for every region, we have 12 numbers for knockout and 12 numbers for wild-type read counts. And the question is there differential read counts between knockout and wild-type, okay? So that we can say that there's differential binding. And we need to do an appropriate statistical test with an appropriate noise model for the read count data, okay? And we use this negative binomial over dispersed Poisson that I understand you've talked about, okay? But we can discuss some more after the break. Okay, so, right, so here we are, we have these events, and we want to know, we have read count evidence here. I'm stacking all the replicates, right? But there are separate numbers, and we're trying to test essentially whether the means are different. Okay, but there's actually an additional factor, and if you ever get into the business of designing new sequencing protocols or using protocols, and you actually have to pay attention, all right? So for example, when you do these, when you design your libraries, there are various linkers and adapters that you use, right? And initially, this 3' linker sequence that we were using a different one in wild type and knockout, and it was just so we would be able to in silico check for contamination between libraries. Okay, so there was a reason we did it, but using different linkers in different samples will create sequence-specific differences in read counts. All right. So we had 6 libraries, and 6 libraries we actually had to do a linker swap and do them again. And then we modeled the linker effect in a generalized linear model. Yes.
Is that because the ligation efficiency depends on the sequence of
Okay, so it's probably ligation efficiency. It could also be that you create these secondary structures sometimes, and it's harder to sequence, right? So, yeah.
And how did you start feeling that it was wrong?
How did we know it was wrong? So, yeah, it was a bit painful. So we saw in the miRNA counts, okay, that we were getting differential expression of other miRNAs besides miR-155, all right? And after following, that's very interesting, maybe, so there wasn't any difference in the cellular concentrations, but maybe they're preferentially loaded, many hypotheses, but then we figured out, yes, it's probably this effect. And when we did the swap and corrected for it, all the differential expression went away except for miR-155. So there's no change in, no significant changes in the miRNAs that are loaded into Argonaut, other than 155 is missing. And we actually, so there was a paper, the PhD student who, MD PhD student who did the experiment saw a paper from Tom Tuschl's lab where they were showing that the adapters give you sequence-specific effects when you do deep sequencing of microRNAs, and there's a way to try to back calculate. Okay, so microRNA deep sequencing, you don't get absolute expression, but you can use it for relative expression, just like microRNAs or microarrays. Right? There was another question.
Do you think that's the case for other adapters that people use in sequencing?
Absolutely, absolutely. Okay, so we'll talk towards the end about trying to do meta-analysis on RNA-seq to probe for these different 3' UTR isoforms. And it's very tricky because if you look at RNA-seq data, your coverage bias, especially over the 3' UTR, is up and down, right? If it were perfect, if they weren't, so I think that's coming from random hexamer priming, right? And if you have sort of big compositional biases, like you have more AT richness in UTRs, it's going to be more pronounced, all right? But absolutely, every sequencing protocol, all the data you look at will have these kind of protocol-specific biases, and it's very difficult to look at one window and another window and say that the read counts are comparable to each other, okay? So all the tests, all the statistical tests that we do are taking one window in one set of samples and comparing to the same window in a different set of samples. Yeah? Okay, so yeah, so we actually had to explicitly model this after accounting for the linker effect. Is there differential read counts in wild type versus non-type? Yeah? Okay, so what did we find? Okay. So about 40% of the stringently differential peaks, okay, so these miR-155 dependent peaks, are non-canonical. Okay, so I say seedless, but that's a little too provocative, okay, because often there's some kind of semblance of a seed just degenerate. Okay, so here's a case where if you take this region that's differentially bound, differential read counts, you get a nice 7-mer, even more complementarity. Whereas here, the best alignment would find a mismatch right in the seed, but sort of the rest of the seed is there. Okay, so not totally seedless, but there's no 6-mer seed, which is the most minimal sequence signal that any computational prediction would use. Okay, so TargetScan, I don't think even believes in 6-mer seeds. They would insist on a 7-mer, length 7 complementarity. Okay, so do they work, these seeds? Do they mediate regulation? Okay, so on the left is the kind of what we do, and on the right is what our experimental collaborators do. So at a transcriptome-wide level, we can say that these non-canonical sites do mediate regulation. Okay, so what we do is we also have expression data. Okay, this is just by microarrays in the knockout mice versus the wild type in these activated T cells. Okay, and what we're looking at is a cumulative distribution of expression changes. Okay, so in black is just the background, all genes. So this is the full distribution of the log expression changes. Okay, if you have a set of genes that are enriched for true targets of miR-155, then when 155 isn't in the cells, you expect them to go up, right? And you expect a shift to the right of this cumulative distribution of that set of genes. Okay. So in green, this is what we sort of facetiously call the bioinformatics prediction. So if you just look for a 6-mer seed in the 3' UTR, you get this kind of mild shift. But it's clear, you know, too many targets, right? There's not 3,700 targets of miR-155. If you look for genes with a single canonical, meaning a 6-mer seed, in a differential binding site, okay, based on Argonaut. So that's in blue, then you get a very robust shift. Okay, so these are, there's the differential AGO binding, the miR-155 dependent binding, and there's a seed, and they are robust targets. But if you look at miR-155 dependent peaks, okay, so differential peaks with no 6-mer seed, okay, and in fact no 3' seed anywhere in the 3' UTR, you get this set. And they're also pretty good targets. You don't have such strong shift at the end, okay, but these are good targets, okay, they're real, at least statistically, yes.
So these were the differential sites that don't contain the canonical seed?
They don't.
All of them? Or did you explicitly look for some variant?
No, so this is all of them. So all we say is there's a differential binding site, and there's no 6-mer seed there or anywhere in the UTR. Okay, so it's a pretty conservative set in that we're saying there's no 6-mer seed anywhere. But with this differential AGO evidence, you do get, you know, so often these non-canonical sites are paired with canonical sites, so it's just a small set, you see, these 32, okay? All right, then Gabe Lieb did experiments so luciferase assays to check these non-canonical sites, okay? So this is just a positive control. SOX1 is a famous known canonical 155 target, and we're seeing that, you know, this double transfection, the luciferase and miR-155, you get 40% repression, okay? If you mutate that canonical site, you get no repression. This is a negative control. Okay, these are all examples of UTR regions with differential AGO binding, but no canonical site. And you get anywhere between 30% and 10% repression. So they work. They're not as strong as canonical sites, but they do seem to confer regulation. They do seem to be direct targets, all right? Backing that up, if you mutate the predicted non-canonical binding site, okay, we're actually mutating what would look like the non-canonical seed, okay? Then in most cases you abrogate the repression. Yeah? Questions?
So the location of the seed matters.
Is the location, you mean in the context of the UTR? Okay, so what's known about microRNA binding, so from the early days, is that you tend to be near the start of the UTR or the end of the UTR. I think is a little more complicated than that, but definitely when you look at these AGO binding profiles, there are islands of lots of AGO binding and deserts of almost no reads, right? And that's some signal for accessibility or secondary structure, that there are regions that seem to get bound by Argonaut, and there are regions that aren't, right? So I think there's, right? I think there's lots to be learned, okay? The little bit of machine learning that we did do, it was modest, but a little bit cute, maybe. Okay, so we needed to summarize what these non-canonical patterns look like. So what we did was just learn the parameters of the local sequence alignment between the miR-155 site and the target sites in a way that the differential sites should score higher than all the other AGO sites that aren't differential. Okay, so it's a discriminative learning. We use an SVM-like algorithm, but we're only doing, in this one, local alignment. So we're only seeing seed-like patterns, all right? And this is really just a summary on the training data. What's nice is without having a special starting point, the alignment learning approach will learn all the canonical motifs. So these are the 6-mer, different kinds of 7-mer and 8-mer motifs that people know about. But then you also see these other more degenerate motifs. So certain positions that seem to allow other mismatches relative to the seed. But you'll also see that only about half, little more than half, of the non-canonical sites are accounted for. So in a separate analysis, we also know that there's complementarity in a region of the, complementary to the 3' region of the microRNA. That accounts for a little bit more, but we still don't understand. We still don't understand, even for 155, all the sequence determinants. Okay. This is just the algorithm we don't have to get into. Okay, so just wrapping up this part. So you'll notice that all the analysis didn't really deal with what we usually think of as the target prediction problem. Okay, so we characterized what's special about 155 sites relative to other AGO binding sites, but we didn't really figure out, given a sequence, how do you tell whether it's a 155 site or not, right? And there are plenty of seeds or seed-like patterns. So every pattern that you see mediating this differential binding, you can see many copies of that pattern where there's no AGO binding. Okay, so there's more that we don't understand. So we are trying to use machine learning approaches on this data set and other data sets to try to learn what are the contextual features, is there a signal for AGO binding? It seems that there is. What are the arrangements of the duplex? Yeah, so that's kind of work in progress. There's also, so if you were looking carefully, you might have noticed that there's something we failed to model, okay, when we were doing differential binding. Actually, can anyone say what we missed when we're just looking at read counts between wild type and knockout? So the thing that we're not modeling is that miR-155 actually affects expression levels, right? And we're not modeling the change in expression. And we can get away with that because when you lose expression of 155, you would tend to get higher read counts, right? The reads mapping to your event scale with expression of the transcript. So it makes it harder to find differential events, and that makes us more conservative rather than inflating false positives. But if we want to look at these more subtle effects, like are there sites that are more bound by other microRNAs when 155 is out of the way, right? Is there increased binding in knockout? Then we actually have to explicitly model in the general linear model the expression change. So we know how to do that now, but we didn't. Okay, so should I? What should I do? I have more. We could go for 15 minutes into the next part.
Let's go for another 15 or so minutes.
Okay. Okay. So so now I'm going to switch to this other project, mapping and quantifying the usage of 3' cleavage sites with 3' C. Okay. So let me get as far as what the data looks like, and then I can tell you the results maybe after the break. Okay. So this is, I don't know, this is an animation by my PhD student, now finished. Okay, so I wouldn't be able to do this, all right? But this is, we're trying to see the transcript elongating. These are, so here's the polymerase making the transcript. These signals are polyadenylation signals, but you pass a whole bunch of them, and then at some point the machinery, the co-transcriptional machinery, figures out that this signal is one that we want to recognize. Various factors come in, perform the cleavage at the poly A tail, and then that's your mature that's your mature transcript that gets shipped out of the nucleus, right? And where this cleavage happens defines how much UTR, how much 3' UTR you get with your message. Yeah? Okay. So our story starts with Christine Meyer, who in her postdoctoral project showed that certain genes seem to preferentially express a shorter isoform due to alternative cleavage and polyadenylation in cancer cell lines. Okay? So what we're looking at is cyclin D1. There are two forms, okay, a long form and a short form. So this is the end of the coding region. This is the last coding exon. And then we have normally this long 3' untranslated region. It's more than 2,000 nucleotides. It contains microRNA binding sites, AU-rich elements, localization signals. Okay? But you can also use this non-canonical, weaker polyadenylation signal and cleave here and get a short isoform that codes for the same protein but has much less regulatory sequence attached for it. Okay? So the APA changes the substrate for post-transcriptional regulation. Okay, and what we're looking at here is Christine's northern blots. Okay, sometimes I see talks by other people, and here's Christine's slide. So she kind of created a monster with this paper that everyone thinks you get shortening in cancer, and that's the end of the story. And what I'm going to show you is it's actually more complex than that. Okay. But what we're seeing, okay, so this is the short, this band is the short form, this is the long form that you normally see. These are different breast cancer cell lines. These are different normal breast epithelial cell lines. These are lung cancer, normal lung, okay? And what you see is that at least in some of the breast cancer cell lines, you get more of this short form that lacks all those regulatory elements. And the part that I don't show here, but is actually quite important, is that often you get upregulation at the protein level because you lose all this regulatory sequence that mediates various degradation pathways. So she showed that relative between the long and short, often, so the short could produce between 2 and 40 times more protein. Okay? So we think of the short as more stable and the long as less stable, but that also is a simplification. we don't really know that mapping. Yeah.
I'm curious that since there's a lot of like inhibitory signals at the certain UTR, and now the only way that is known to try to short this site, but there are other known mechanisms, for example, a protein that tries to bind to a certain UTR to protect the messenger from macro.
Okay, so other than, okay, so I don't know about protection from microRNA, but there's probably a lot of localization. So maybe, you know, the long sequences can go somewhere in the cell where they're protected from everything, but they still are stable, and then they're used later and can be regulated later. So I think it's more complicated. The story people think of is, you know, you lose microRNA binding sites when you get this shorter, and then you can be expressed higher. But again, I think that's an oversimplification. Okay, maybe an appealing one, but it's an oversimplification. Okay, so since that paper, there have been many studies, or you know, some predate, I guess, the paper, looking. Trying to look globally at this relative usage of short and long 3' UTR isoforms. Most of these studies used microarray technologies, exon arrays. So there you're hoping that you have probes in the 3' UTR that give you enough resolution that you could see differential usage of these, differential expression of these isoforms. Very, very tricky. Okay, so some of them were meta-analysis of microarray technologies. There were also early, or 3' sequencing, 3'N sequencing technologies. All right, and I don't want to dwell on these findings because again, I think it's some of them were very early before you had good data. And some of them have been overgeneralized. For example, if you see a recent review in Nature Genetics Review, overgeneralization of these results. Okay, so I think we have a more complex picture from our data. Okay, but what people have found is that at least in certain physiological activation states, so the T cell activation, synaptic activation. So there's some tendency to use more of the shorter isoform. Again, this was a microarray analysis. Also oncogenic transformation, some tendency, like in Christine's data, to use the shorter isoform. And in vitro differentiation from mouse ES cells to the neural lineage, you get lengthening. Okay. All right, so so when we started collaborating with Christine, we quickly realized that we couldn't really address this problem using meta-analysis of RNA-seq data or, God forbid, array data. We needed a quantitative sequencing protocol to map the 3'Ns of transcripts. Okay, so this is a tag-based sequencing protocol, right? So we're just trying to get a read from the very 3' end of the transcripts, and we're trying to get them actually in a kind of peak region up to the very end. All right, so we think that getting some range, but up to the very end will give us a more quantitative signal. We also don't use any random hexamer priming, so we really want we want to capture the 3' ends and treat them all the same in the protocol. Okay, so right now the protocol that will be published, but it's still being developed, so it's still evolving, involves a pull down with a poly-DT feed. Okay, so this is the poly A tail of the transcript. This is the part you're interested in. Okay, we have a trick or several tricks. So this anchor sequence that's not all T's is so that you encourage it to anneal at the very end of the poly A tail and not in the middle, so you don't get internal priming. And we
this
also
is the V that we talked about, the IUPAC symbols, right? And not T symbol, just to remind you guys.
yes, you can probably tell me better than I can what V stands for. Okay, but our other trick is this ribo-U that we'll get to in a second. Okay, so we do RT and second strand synthesis. So now we have double-stranded cDNA, and now we use this ribo-U. We can introduce a nick here, so we can do a cut, and then we have an enzyme that will do nick translation. It just pushes the cut. It's a timed reaction, so we can push it 50 to 75 base pairs here. Okay, now we cleave at the nick, add the second adapter, and sequence in towards the poly-A tail. Okay, so this is the sequence we get. Okay. Yes.
I don't understand that nick translation step. Can you give a little bit more detail about how does this X move?
Yeah, it's a different polymerase, right? It's just repairing and then eating the next one, right? And so after the timing, you have a nick here instead of here. So it's just pushing the nick along.
Oh.
Okay. I forget.
It's like ligating those.
Yeah, so it's a different polymerase, but not Pol2.
Kind of moving to the right. But the nucleotides themselves stay.
Yeah, they stay there, right? So this is double-stranded. So you're just kind of at the end. So after a certain amount of time, the nick is here, right? And then it repairs that, and yeah.
So it's a processive enzyme, but it doesn't
yes.
do the NTPs, right? It just works with the ones that are already there.
Yes, yeah, but yeah. So we end up with the nick out here, and then that's how we get the piece of the end that we're interested in. Yeah. Okay, and it also treats all the 3' ends similarly, right? Yes. So this is it's another enzyme, and it's not great if you have this blunt end and then doing the ligation of the adapter, but that's what we have. We have a blunt end, and we're ligating an adapter here. So it's just every step cuts. Yeah, yeah.
So what you're sequencing in the end is just that 50 to 75 base pair region?
Yeah, so we're doing 50 reads, and we're getting, so you don't want to have a lot of A's. Okay, other protocols sequence out from, like they try to sequence into the transcript from the tail, but then in the cDNA step, you have to prime it with random hexamer primers, and maybe causes it to be less quantitative. Okay, so here's what the data looks like after some cleanup. Okay, so what I'm showing you is the end of the gene models. Okay, so this is the last coding exon. Here's the 3' UTR. And what you see is, I'm showing two different cell lines, MCF7, MCF10A. Okay, sometimes you just see one peak. Sometimes you see two peaks. Sometimes you see three peaks or more sitting over top of the 3' UTR. But what we like is that it seems that the read coverage is quantitative and correlates with northern blot analysis. So if you look in MCF7, you have these two peaks, but you have much more of the short isoform than the long. Whereas in MCF10A, you sort of have more equal coverage, right? And here in the northern blot, here's the long and short, and here in MCF7, you see much more of the short, right? And the positions look right, but also the relative read counts look like they're quantitative and correlate, okay? Which isn't true of some of the other 3' sequencing protocols. Okay. Yeah, okay. Should I? Okay. Yeah. So I mean, okay. Okay, so the first question is, what is the nature of variation of these 3' UTR isoform? Like, what's the nature of variation of expression over different human tissues? Okay, so there have been papers using 3'-seq on different tissues before, in mouse and human, different cell lines, but it never really tells you anything other than there's a lot of alternative cleavage and polyadenylation. Okay? So what we find is that the events themselves are constitutive in the sense that they're always occurring in the same place across all the tissues. But the expression is tissue specific, the relative expression of the isoforms. Okay? So, for example, if you see T21, there seem to be 2 isoforms. Testis is very short, okay? So you see a lot of the short isoform and very little of the long, but you still see some of this long. In brain, you see a lot of the long, but actually there's still a little bit of the short, okay? And in general, you see both isoforms, but their relative read counts change, okay? Here you actually see the same 1, 2, 3, 4, 5 isoforms. You do see isoforms beyond the ref-seq annotated 3' UTR end, okay? But essentially, it's not true that every tissue is inventing a different place to end the transcripts. They're mostly the same, and the true cell-type specific APA events are rare. Okay, so maybe it's a banal point, but that's what we see. Okay, and I don't think that was known. This 3'-seq and tell you what we think is the story for this data set, and then I think you've already you've already seen a lecture on over-dispersed Poisson distributions and some of the analysis, but I'm gonna do a sort of brief recap about that and talk about the generalized linear models that we use for the analysis in this whole talk. So some of it is review, but maybe with a slightly different point of view, but and definitely a different application. So not straight RNA-seq differential expression, but this differential usage of events. Okay. All right. So where we were before dinner, we had said that when we look at these events across many tissues, what we see is that the events are actually a particular gene has the same number of events and positions, but just the relative read count evidence, the relative expression of these 3' UTR isoforms changes with tissues, and that's the variability that we see, rather than seeing a particular event only in one tissue, truly there, high, and never in any other tissue. Okay, so since we know that is true, we can talk about single isoform genes and multi-isoform genes, or single UTR, multiple UTR genes. Okay. All right, and we can also talk about regulation of these events, or differential relative expression of these isoforms. Okay, so when I showed you northern blot analysis, I tried to convince you that this protocol is relatively quantitative. So usually with read counts, you wouldn't compare read counts in this event to read counts in this event. And for the statistical analysis, we don't do that. But for visualization and some of the qualitative analysis, we talk about this. Oh, sorry, I've shown this enough times. This 8 should be here. Okay, so we talk about this short UTR index. It's just what fraction of reads, of all the reads mapping to the 3' UTR of a gene in a particular tissue, what fraction go to the shortest isoform? What fraction are at the most proximal polyadenylation signal, okay, most proximal to the stop codon, okay, as opposed to the more distal ones. So this one is, in this tissue, maybe 80% of the reads, 0.8, whereas in this tissue, maybe 0.3. And this is a pretty big change in this short UTR index. And we think, a change of about 0.2, 0.3 is detectable by northern blot analysis usually, okay? Now that's not how you should do the statistics, okay? And I'll show you how we really do it using a generalized linear model to test for differential 3' UTR isoform usage in a little bit, okay? 2 slides, okay? But let's just think of, if in a tissue you tend to express more of the short isoform, then your SUI is high. If you tend to express mostly the long isoform, the SUI is small. Okay, so not much of the short, mostly long. Okay, and then if you just okay, this is one visualization. Probably I should show you the distribution of SUIs over all the co-expressed genes, but this is just showing the median short UTR index, right, as a kind of just a metric of how short is a particular tissue, okay? So testis is short, brain and ovary are long. ES cells are actually somewhere in the middle, okay? And this is just to show you that these truisms that we've started to accumulate, that as you differentiate, you get longer UTRs in general. It's not true. Okay, so if you look at undifferentiated versus differentiated, it's sort of across the ES in the middle. B cells are short. Okay. Breast and other epithelial cells are long. Question?
Just to make sure I'm understanding correct. So in the cases in which you have several peaks,
right,
it means that you have many different lengths, right?
When you have 2 peaks, okay. When you have 2 peaks, it means that you have a population of cells, and every cell has a population of transcripts, right? And we're finding, we're detecting expression both of transcripts that have a short UTR and transcripts that have a long UTR. But they're really, if we see 2 peaks over the UTR, they're really just 2 different isoforms when it comes to the 3' UTR. There could be other splicing that's invisible to us, right? But in terms of the variation that's generated by alternative cleavage and polyadenylation, there's just 2 isoforms. And because the method is fairly quantitative, right, if we see mostly the short isoform, that means in that tissue, we're seeing high expression, relatively high expression of the short isoform and relatively little of the long isoform, okay? And it's, as I'll show you in a minute, the genes that shorten or lengthen in different tissues are tissue specific. So this is just summarizing. And when I say this shortening and lengthening, I don't mean that literally. I mean a shift towards higher expression of the short isoform versus a shift towards, right? I don't mean actual trimming or anything, right? Just these 2 isoforms and relative usage. Okay, so this is very crude, but even at a very crude level, you can see it's not true. Okay, so it's not true that as you differentiate from ES cells to any differentiated cell that you will always express overall longer UTRs, because B cells are differentiated cells and they actually have really short, relatively short UTRs in general, the median. Okay, it's also not true that in cancer versus normal cells, you're always shorter. okay? And we're investigating this in a bunch of different ways. It really depends how you do the comparison, but there's some cell type specific profile, right, that we're getting at, and depending on the origin of the cancer, maybe you can shorten or lengthen relative to the original cell type. So we haven't got there yet, and we don't fully understand, but this is just showing it's not true that every cancer cell line is shorter than every normal cell line, okay? And it's also not true necessarily that the cell types that proliferate more have shorter UTRs, okay? So that's another thing that's been in the literature. If you look at cell lines versus the tissues, okay? So all these, it's more complex, yeah? All right, so how do we really test for significance of differential usage? Okay, so as I said, we're going to revisit this in a little bit. Let me give you the flavor of what we're doing. So we adopted, altered slightly, but we're using an approach that was developed for detecting differential exon usage for alternative splicing from RNA-seq. Okay, and the idea is you have these different events, so exons in differential splicing. For us, it's these cleavage events that we have read count evidence for. And what we want to know is their differential usage of the events after you correct for differential expression. Okay, so here's what it looks like, and we're going to come back to this. Okay, so I just at a high level, we're modeling the log read counts for an event. Okay, the mean, the expected log read counts for the event, as a linear model depending on these factors. Okay, so we're saying maybe there's some baseline expression for the gene. Okay, so this is like your intercept. There's the log fraction of reads that we expect on average to land in this event. Okay, so maybe it would be on average 60%, and then here 40%. There's a factor that allows us to look at condition-specific gene expression change. Okay, so maybe in condition C relative to the reference condition, we have an expression change. And then there's an interaction term between the condition and the usage of the event. Okay, so is it true that I just basically have 40% of the reads here, 60% of the reads here, and then it scales with expression? Or is that not enough, you know, like baseline expression, baseline usage, and then condition-specific expression change? Is that not enough to describe what's happening, or do I need an additional term that allows, with certain conditions, I change my log mean? Yeah.
So would that be because of the different protein combinations binding to the mRNA and to the secondary structure,
Right.
that kind of stuff?
So, okay, so this is just a statistical test to say, is this interaction term 0 or not? If we need this interaction term, it says that the event somehow, or the expression of that 3' UTR isoform is somehow regulated. So just a word of caution, right? So two things can change our read counts. There could be regulation at the level of the choice of event, the cleavage. So you could just be making more of the short or more of the long. But then there's also post-transcriptional regulation. Maybe you make long and then it gets degraded.
It's just
and
an artifact knows what you're after.
This is, until you need different data if you're just going, sort of the nascent transcript or something. You need to measure something else if you want to isolate just the effect of the APA and not look at the post-transcriptional regulation. So we're just asking right now, statistically, do we have events like this where it seems that there's a statistically significant change in the relative usage of the events? And this is the test. So we have this generalized linear model, and essentially we're testing whether an interaction term is 0 or not. Okay, and we have I know the notation is a little bit heavy, but we have these interaction term. We have a model. You have a model, a smaller model with no interaction terms, and a bigger model with the interaction term across all the conditions. And you're asking, are these all 0 or are they not all 0? Okay, so it's the equivalent, like conceptually, of an F-test, right? But it's not an F-test. Okay. Does it make sense? So we will come back to this, but this is the test, and this is using the over-dispersed Poisson, the negative binomial model for the read counts. Okay. So we'll get back to this, but just visually, okay. So here's an example of a regulated event. And it means that you need that interaction term in order to model it, right? You do a statistical test and you say, well, the model that has the interaction terms is better than the one where it's all zeros. And you get a highly positive coefficient for interaction term here in this tissue and a highly negative one here. So you might have some mean fraction, mean usage, but it's very much, you get much more usage in testis than on average, and much less in brain on average. Okay, so we have this coefficient, and the size kind of tells us something about. We have a p-value for whether it's 0 or not, and the size of the coefficient tells us something about more usage, less usage. Okay. We'll come back to it. Okay. Yes. Okay.
Is it by construction? I guess there's a peak at the end of the 3' UTR, right?
Okay. So.
But then whether there's one at the beginning, that's kind of dependent on the gene.
Okay. So we're only testing multi UTR genes. So you have to have 2 events for there to be anything to test. And if you've played with these linear models, you know that. Okay. So. I have an event here and I have an event here, but they're not independent. There's not really 2 things to test. If I test this, then this is also going to be significant. And in fact, basically, I have event 0 and event 1, right? So I don't need 2 coefficients for this. I really need just 1 coefficient. Am I the first event or am I not the first event? Similarly, across conditions, I have to do it relative to some reference condition. So if I have 7 tissues, it's really 6 coefficients I'm talking about. Okay, so there, yeah.
So I'm confused about maybe you could the way you drew it. Why are there no reads in the middle of the
UTR? Oh, there aren't any reads in it. So because this is 3' seq, right?
Oh,
right. So this is an art. Yeah, we are cleaning up a little bit. Okay, so we have some. So you still get internal priming. So if you have a region of lots of A's, right, that can get pulled down and seem to look like it's a 3' end when it's not. It's in the middle of a UTR. So if we don't see any kind of polyadenylation signal, but we have really a big A, we throw it out. Okay, it could be real, but we're conservative and we throw it out. If we have an event with 1 read or with very low read coverage, we have a tags per million threshold, right? Because if you have an event that is used 1% of the time, is that alternative cleavage and polyadenylation, or is that noise, right?
You
so,
can do this with paradigms too. You don't need to use
so so you could use paired end in our protocol. We just have single end. But what I would say is it's difficult to do this with RNA-seq. Okay, we tried, right? So if you're expecting that you have read coverage and maybe you see a change point in the read coverage, like you have high reads here and then low reads all through here, right? Because this is high and this is low. It's very, very hard to detect that without knowing where the 3' ends really are, okay? So I'll show you at the end that we are doing that meta-analysis, but we know where the 3' ends are. If you don't, I think you will get a lot of false positives due to coverage artifacts, okay? Okay, so for now, let's just leave it as we have a statistical test that can say whether, okay, I think in the paper, the manuscript, we're calling it polyadenylregulated, okay, not my idea, but you know, is the expression level apparently regulated across, the relative expression regulated? So is there a shift, right? Or is it not regulated? It's multi-UTR, but it's kind of the same thing all the way through. And we have a test for that, okay? So polyadenyl regulated PAM genes, so regulated or not, okay? So we can make that call. Okay, so, okay, and then we can also do pairwise comparison, right? Does it look like this gene is shorter in testis than in ES? Is it shorter in B cell than in another tissue? Okay, so we can do pairwise comparisons with the same a similar statistical test, but just two tissues at a time. Okay, what do we think is going on? We think that maybe alternative cleavage and polyadenylation is a way for ubiquitously expressed genes to reinforce tissue-specific expression programs. Okay, so why we think this is that, okay, so as we saw, you can, you know, wherever the gene is expressed, if it's single isoform, you just see one event. If it's multiple isoform, you see the same events, okay? So we think that this single isoform, multi-isoform is fairly stable regardless of how many tissues you see the expression in. And what we see is that if you are expressed ubiquitously, say in 6 out of our 7 tissues, you're fairly likely to be multi-isoform. So more than half the genes are multi-isoform, whereas the tissue-specific genes, they seem to be single isoform, mostly.
I have the sense of, perhaps you said it already, how many is the number? Like, how many things are we
How many events? Okay, so in this atlas with the 7 tissues that, the 7 tissues, the analysis is about 15,000 transcribed genes and about 37,000 events that land just in the UTR or, you know, an extension of the UTR. There are other events that are upstream, antisense, intergenic, intronic, okay, but just these UTR events, about 37,000. And these are fair, you know, real, not 3 reads, right? I can go through the actual filters that we use, but it's just, we want to see things that we think could be real in a tissue, right? And if it meets some sort of threshold in a tissue, then it's likely to go in. Okay. So what we think is the tissue-specific genes that are narrowly expressed, they don't need APA. Okay, you can regulate them transcriptionally. They're on in certain tissues, off everywhere else. But the genes that are expressed across many tissues, maybe they can use this. And in fact, more is true as you increase the breadth of expression, you're more likely to be multi-isoform. the genes that shorten, that are ubiquitously expressed but shorten specifically in a tissue, seem to reinforce or have it be enriched for annotations that are associated with that tissue. Okay, so all the genes in this diagram are expressed across all our tissues, but what we're showing, if it's red or white, it means it's shorter in that tissue. So it's a short UTR index. Blue means it tends to be longer. So you have a bunch of genes, a large number of genes that seem to be short in testis. You have other genes that are specifically short in B cells. They are associated with lymphocyte activation, regulation of NF-kappa B. Okay, some genes that are short in muscle or ES. All right. So yes.
I'm sorry. And the columns in the
I'm sorry. So this is actually, so the rows are genes and the columns are tissues, but I haven't the first is testis. Okay. The second is B cell. Yeah. Sorry, it's poorly labeled. Yeah. So this is just showing. So to get into this picture, you had to pass a statistical test that you were shorter in all the pairwise comparisons compared to other tissues. Okay. All right. So all right. So leaving aside the tissue-specific changes for a second, we also, this is getting cropped at the bottom, but we also wanted to look in isogenic cell line systems where you just do one change. Okay, so I guess you do one change, but it's a transformation. Okay, so we're looking at starting with B cells and transforming with Epstein-Barr virus, so transforming into lymphoblastoid cells. And this is MCF10A, where we're just overexpressing one oncogene, HRAS. So it's an oncogenic transformation. And what I'm showing, these are, if you've looked at EDGAR or DSeq, you might have seen these kind of plots. Though I'm actually showing the UI, but the statistical test, again, is not based on UI. Okay, so as a function of the log expression, these are events that are genes where we get shorter in after transformation, longer after transformation in the B cell to lymphoblastoid cell, the EBV transformation. This is shorter after overexpressing HRAS and MCF10A, longer. Okay, so in both cases, you do have more shortening than lengthening, but you actually have both. Okay. But the important thing, I'm not sure this is the best way to represent it, but if you look at these genes that are changing in the non-oncogenic transformation, and you put, where are these genes that changed in the oncogenic transformation, they're different. So I'm putting these genes and highlighting them over here, and I'm putting these genes and highlighting them over here. So the genes that are changing, that are shortening and lengthening, are specific to the transformation, to the stimulus, and not just a proliferation signature or a So we think it's more complicated than just proliferation or transformation. Okay. All right. So then we wanted to look at independent kind of evidence that these classes of genes that we're defining, the genes that are multi-UTR and regulate the isoform usage versus the ones that don't, are they actually different functionally, architecturally? Okay. So the first thing is that the genes that are regulated at the level of APA, they actually participate in different processes than the ones that are just they don't regulate. You just see the same ratios across the board. Okay, so you have annotations. These regulated ones are involved in ubiquitination, regulation of transcription, a lot of transcription factors, RNA processing, also transport. Whereas maybe these others, it's not such a clear enrichment. The single UTR genes definitely sort of housekeeping like annotations. Okay, protein biosynthesis, energy metabolism. Okay. The single UTR genes have short UTRs. Okay, so it's not, that's not a priori obvious that that's going to be true, but the single UTR genes, they're short. The regulated multi-UTR are longer, and the not regulated multi-UTR are sort of in the middle. Okay. If you look at the single and the multi-isoform genes, where the red line is where the first proximal cleavage site is. So it kind of looks like with this unregulated multi-UTR, the first cleavage is kind of where a single isoform one would end, and then you have more UTR on another cleavage site or more cleavage sites. These regulated multi-UTR genes, they are longer, and they also tend to lose more of their UTR when they use the most proximal cleavage site. Does that make sense? Okay, so this color code is trying to be consistent. If you look at the range of the short UTR, so these regulated ones, we expect there's a big range. Sometimes the proximal cleavage event is always used, sometimes it's not used, or in some tissues it's highly used, some tissues it's not used, okay? So you get this big range. These non-regulated ones, they are just long, okay? So very little usage of the proximal, lots of usage of the distal. Okay, that's what we see, right? Again, it's not obvious that we will see that, but across the board you see long and a little bit of short. Yeah? Okay, but what's not true is that, so these multi-UTR genes, usually this proximal cleavage event, it's kind of weaker, right? So, but there doesn't seem to be a difference. So, you have less use of the canonical polyadenylation signal, more of these less used ones, but these proportions seem to be the same between the regulated and unregulated, all right? So we think, maybe there's a model that you start out as a single UTR gene, and then as you get expressed in more tissues, somehow you acquire this other, a more distal site. You have to make this weaker, and you make this longer, but then somehow you also, for some genes, you regulate this weaker site so that sometimes you can really use it, and mostly you don't use it. Sometimes you use that proximal signal, and we don't fully understand. Okay. One more slide just on the big picture. Okay, so what about microRNA binding sites? Okay, so it's tricky, and we went back and forth many times, separating cause and effect, because if you can regulate your proximal site so that sometimes you include the distal part, sometimes you don't include the distal part of the UTR, right? Maybe the whole signal you're seeing is just that in certain tissues you're getting degraded by microRNAs, tissue-specific microRNAs. Okay, we don't see that signal, we see a slightly different signal. Okay, so if you look for what microRNA signals are enriched in the distal region of these regulated multi-UTR genes versus the proximal, okay, they tend to be ubiquitously expressed microRNAs. There are some exceptions, like miR124, that's a sort of brain specific, but often it's ubiquitously expressed microRNAs sites that are enriched in that distal part. And the enrichment is not true for these unregulated genes. For single UTR genes, that single UTR tends to be there's some enrichment for tissue-specific microRNAs. Okay, all of this is very subtle. Okay, what I'm doing here is doing a null model of scrambled microRNAs hitting scrambled sequences hitting conserved regions, and I'm doing an empirical p-value. So, you know, we're trying to control for sequence composition bias, for conservation, etc. Okay. The other thing that's true is that these regulated genes, these regulated genes, they tend to be more conserved than the unregulated genes around the proximal site, though also around the distal site. I'm not showing you that. Okay, so there's some model here. You know, you have these regulated genes, they're really long. They can lose a lot of their UTR. This part that they use, that they lose, are enriched for ubiquitously expressed microRNA binding sites. Okay. Whereas the single UTR genes are short, they are somewhat enriched for tissue-restricted microRNAs. Okay, so I think there's still lots to learn, but that's what we came up with from this tissue atlas that hopefully will be published soon. Okay, yes.
Does it change, does the analysis change when you retest this out? Is that just a weird outlier?
So testis is an outlier in, like, it's very short, okay? And I guess it's also a model system for post-transcriptional regulation. So maybe it's short because there's a lot of post-transcriptional degradation. We don't know that it's choosing the proximal site. But if you took testis out, then B cells would be the outlier, and the rest of the atlas, they're quite short. Okay. All right, so something we're working on. So as I said, we actually started with this project looking at RNA-seq data, and back then, definitely, there was very little statistical traction trying to find change points in RNA-seq to figure out, is there differential isoform usage. But if you have an atlas and you know where the 3' ends tend to be, right, you can imagine various schemes. Okay, here's one where I know that there's an end here and an end here, and I could put a probe right at the end, and then I could do a DX-seq-like analysis to say between normal and tumor is the differential usage. Okay, we actually aren't doing this. We're doing more like a change point version. Okay, but we're putting a window before and after the proximal or internal cleavage signal, cleavage position, and we're doing a test. So again, with the right read count statistics, we can probe for these changes, and that seems to work. Okay, so here's one. You know, if you look at this was RNA-seq from TCGA. We didn't. We only had normal brain from the human body map, so there are no normals in this study in TCGA. But we're looking at one example of so colon 3. These lines are known from our atlas poly A cleavage choices, right? And what you see is this one seems to be regulated, that you see this longer transcript in normal brain, but not in the tumors and not in these kind of tumor spheres that are related to the tumors. Yeah, so you can get, it seems, some traction. We're still checking this out though. Okay, so this is a breast cancer story. Again, we're showing something that's kind of like a pseudo short UTR index, okay? But just for visualization purposes, again, we're doing the right statistical test. This is breast tumors and matched normals. We're not using them as matched normals, we're just using them as two classes to do a statistical test. And the lines here are genes where we see sort of more usage, more shortening. So in these solid epithelial tumors, we do seem to see shortening. Okay, but we don't think that's true for all cancers. Okay, but this is still in progress. I think we're, the next step, we're just generating RNA-seq and 3'-seq in the same samples so we can check ourselves how good the test is. So I'm still, it's a pretty tricky business. You'll have some samples that seem to have very weird coverage artifacts, and those will be flagged by your, your test, and you just don't know, is it real or not? Probably it's artifactual. Okay, the last thing I can tell you about research-wise, so the other thing we're doing, bringing these two projects together to some extent, we're actually interested in the cell type specificity of microRNA-mediated regulation. So I told you back before the break that miR-155 is actually upregulated upon activation of many immune cells. And we also know that APA could generate different relative, so it generates different UTRs and different relative expression of the UTRs across the tissues. But what we can do is in our mouse model, right? So in a wild type mouse and in the miR-155 knockout mouse and in different cell types, we have 3'-seq data, so we know we can look at relative expression of different 3' UTRs, right? Different 3' UTR isoforms, and see if, even for the same UTR in different cell types, is it regulated by miR-155 to a different extent? And we have some preliminary evidence that that's true. Okay, so even when you control for you're looking at the same UTR, a microRNA can regulate its target to a different extent depending on the cellular context. Okay, other RNA binding proteins besides Argonaute, other microRNAs that are expressed. Okay, so you could imagine how this could be true, and it seems to be true, and we're trying to decipher that next. Okay. Yeah. So yeah, we're doing a lot of machine learning on our data set. What defines a functional polyadenylation site? The machinery passes all these things that look like they should be, perfectly good polyadenylation signals, but you can learn the other contextual signals that are needed to, that they're always skipped. And then what defines a regulated versus non-regulated site, or the tissue-specific regulation? What are the additional signals that are there? If there are indeed signals in cysts, that might not be the whole story. Right. How have APA and microRNA-mediated regulation co-evolved? All right, so there's probably an interesting evolutionary story, but we're...
Was it like maybe some evolutionary escapism going on?
So we think it's the other, is it that they want to, do transcripts want to escape microRNAs, or did they want to be regulated? We think they want to be regulated, but it's very hard to it's hard to pin down that question. Just that as you become expressed in more tissues, then you somehow gain. Maybe you need to gain signals that will allow for regulation, right?
Yeah.
So
that's great because you can hedge your bets, right? Because you can always go back to the short isoform, right?
Yeah. So yeah. So I think it's very difficult to tease cause and effect. I think maybe we need data where we're seeing just the nascent transcript, and we can really separate APA signal choice from post-transcriptional regulation. Okay, and let me just thank all the people who did this before switching to the other talk. Yeah, so Ali Khan he was a PhD student in the lab. He's now a postdoctoral fellow at University of Chicago. He did all the CLIP-seq analysis. Steve Leonoglu, great PhD student who just defended, and he's now a computational biologist at Genentech. The rest of the people in the lab, Sasha Rudensky and Christine Meyer were the collaborators for these two projects. Okay, so maybe we'll try to wrap up by 8:15. I think so. This is an intro to differential read count statistics. We developed this for a course that Olivier and I and another person co-taught, and I think you've seen some of it. So I'm going to just go through the over-dispersed Poisson part more quickly and get to the generalized linear model part. Okay, so in all these problems, okay, the problem is we have, we're looking at read counts at a genomic locus, okay, so we'll call it, or in our case, transcriptomic locus, all right, but we call it a counting bin. Okay, so we have a window and reads fall into it and we count in different samples. And generally the big question is we want to know between two conditions, when do the counts differ significantly given the biological variation between replicates? Okay, so given the level of variation between biological replicates, can we say that the read counts are significantly different? Right? Yes.
So which are, or this is more general, not just the APA-seq.
Yeah.
I don't think it's a single position of the RNA, right?
Yeah, so I mean, the APA stuff and also the ClipSeq stuff, we're actually going to need the full, like, generalized linear model part. We need multiple factors to explain, okay? But the most basic idea is this counting bin idea, same bin, multiple condition, or two conditions, biological replicates, all right? You can't do the analysis without some kind of replicates some kind of biological replicates to estimate this variation, okay? So you can do differential expression in RNA-seq data. So your counting bin, typically people will use all the coding, like the exon bins, the coding region, but you could use the whole locus if you wanted to. Differential binding events like ChIP-seq, ClipSeq, we use it for DNA-seq. We use these differential read count statistics for, all these kind of binding event assays. Alternative splicing and other kind of mRNA processing, differential usage of exons or 3' ends or 5' ends. There are a lot of applications, okay? So we already saw, I'll just go quickly, one case. This, your counting bin would be a peak event, right? So we identify this peak and here's the event, and then we have the read counts in the wild type and the miR-155 knockout, and we have all the replicates, and we want to know, are the read counts different between the two conditions, okay? And I also explained why this isn't a simple application, okay? We actually have two factors to control for, okay? So we have the condition, so wild type versus miR-155 knockout, but then we also, because of this technical detail in our sequencing preparation, we had two different 3' linker sequences, okay? So we had 6 replicates for each of these pairwise choices. So 12 wild type, 12 knockout, but of the 12, half were one linker sequence, half were the other, okay? So we have to model this linker effect, and after we model the linker effect, we can ask, is there a significant read count difference between conditions, right? Okay, and we've just spent quite some time talking about this APA, all right? And here, it's this problem, okay? So if you look, these are some data that we don't put in our paper, but you could imagine different conditions, we have these different events, and we want to know, is an event regulated, so that the relative read counts mapping to this event compared to all the events in the 3' UTR, is it changing after you count for differential expression? Okay, so this, if you count up all the reads here, it's high, this gene is higher expressed than here or here, I think. I think that's true. Oh, maybe not, because it's a different scale. Okay, there's differences in expression, but after you control for differences in expression, is there also difference in relative expression between the events? Okay, and for that, we're going to need these multi-factor kind of design. Yeah? Okay, and then finally, the differential splicing. The counting bins are the exons, and this is an example from DXseq where they're looking in Drosophila. They knock down some splicing factor called Pacilla, okay, and they're trying to see, are there exons that are differentially included after you control for change in expression, okay? All right, so I'm going to just quickly talk about these basic ideas in modeling count data. I think this is a review. You can tell me if it's too slow. And then the GLM stuff. Okay. Okay, and I'm following, most of this is from Simon Unders, who's the DESeq author and DXseq author. Yeah, so the other packages at JAR that you've already used, you know, we use them both. Okay, they behave slightly differently. Okay, so some of the basic problems with, or challenges with next generation sequencing data, right? So if you look at the distribution over your counting bin, the distribution over replicates, right, of reads in your counting bin, it's discrete, so it's count data, it's positive, it's skewed, it's not log normal, right? So if you make the assumption that it's log normal, especially for these smaller events where you don't have a whole, like all these exons, you're making the wrong assumption, it just won't work. Okay. You tend to have a small number of replicates. You have different sequencing depth in different samples. Okay, so you need some kind of normalization by scaling factors. There are various tricks to do this. You have a large dynamic range, maybe 5 orders of magnitude, okay, and heteroscedasticity, so you have a different variance for different subpopulations of genes or events, okay, depending on their expression level. Okay, so, okay, the normalization, that's fairly easy to deal with. The main thing, and I saw your slides that you've already touched on this, you shouldn't be dividing by total library size. So this kind of tags per million or RPKM, that doesn't work because certain genes that are highly differentially expressed between conditions can skew the statistics for everybody else. Okay. So what you tend to do, so this is the DESeq solution. Okay, you create a reference sample where the counts for each event are the geometric mean of the counts you saw across all the samples. And then you have a scaling factor for every event, the sample versus the pseudo-reference. And then you take the median of those scaling factors. So not the outliers, right? There are counts that change a whole lot, but the median, and that will be the scaling factor for the whole sample. Okay? So that's one solution. It's just to correct for these outliers that change a lot, and a lot of the sequence, a lot of the reads could go to some highly upregulated gene, and that skews it for other genes. Yeah? So you saw this, right? Yes. Okay. Right. Okay. So, and you probably also saw, okay, so the first papers that came out and were trying to do some statistics on RNA-seq and next generation sequencing data made a Poisson assumption. Okay, so you think count data Poisson is reasonable. I just want some notation here. So in a Poisson distribution, the mean is the same as the variance. Okay, we'll call it lambda. And I also want to define this coefficient of variation. So the standard deviation divided by the mean. So it would be 1 over the square root of lambda here. Okay. So how much does, yeah, the ratio of the standard deviation by the mean. Okay, sometimes I'm going to plot the square of this squared coefficient of variation. So sigma squared over mu squared. Okay. Okay, so the first thing is that the Poisson model underestimates the variation between biological replicates, which I think you already saw, right? So what I'm doing is, you know, this is just, think of every point as a read counts for a gene, okay, or an event. We're looking at the mean read count across replicates and the variance across replicates, log, right? If it were Poisson, we should see that they're equal, right? But they're not. It's the variance is higher than the mean, right? Similarly, so if you make a Poisson assumption that the mean is the same as the variance, you plot them, you should get this linear, this equality relationship. If you look at the mean versus the squared coefficient of variation, you'd expect this fall off, this 1 over lambda fall off. Okay. But it's not quite that. It's more like one of these. Okay. So if you say, okay, there's some over-dispersion, that the variance is the mean plus some alpha times the mean squared, okay, then you track the data a bit better. Okay, so that's the dotted. Okay, you track the data a bit better. Or you could be more general and say the variance is the mean, that would be the Poisson, plus the over-dispersion is some function of mu squared. Okay, and you could fit a functional form and get this. Okay, so that's, you know, that's the basic idea, that you need something like this to really capture the variation you see in biological replicates. Yeah?
So what's going on with all the points under the line? Is it under dispersed points? Why are there so many points there?
Yeah, you know, there's scatter, okay, and however you plot it, there will be your fit, right? For some, so what DE-seq does is it fits an alpha, a dispersion function, as a function of the mean by a sort of functional form, and you will always see points that are more dispersed than your fit and less dispersed than your fit. And what it, you know, based on just the sample variance or the, like, yeah. So what DE-seq does is it bumps things up, like it always increases the dispersion, right? So it'll bump it up to the line if it's under dispersed, and if it's over, it'll keep it the way it is. Okay, so it'll make the conservative assumption that, you know, we're going to share across all the events, but we're always going to inflate beyond sort of what the sample would tell you about the dispersion. Yeah.
You figure B, the blue line, is it highly affected by the no level count, the genes with no constantly, for example, below 10? If you filter this out,
So so it's true. It's true that these this, you know, like you have the most dispersion at the low expression level. But I mean, actually, Anders sort of explains it in a different way. Let's go through it. So it's not true. It's not true. It's not true that if you throw away the really low events, that you don't have to worry about biological variation anymore. Okay, so it's not true. This is another of these. You know, this was a study sort of. Again, showing that the variation that you see between technical replicates, so, you know, same sample and you run it through the sequencer several times and you get this, you know, that looks more Poisson, okay? That's kind of what you would expect, but the biological replicates are the ones that are over-dispersed, okay? So, the way Simon Anders explains it is, so you have shot noise. He says, okay, that this actually dominates for the lowly expressed genes. So shot noise is just this theoretically unavoidable. You're sampling from counts, and you will get variation in the read counts because of that, even with perfect replication, right? So this is unavoidable Poisson shot noise, and you can say that that's the variance will be the mean for that noise, and that dominates for the low. It's really bad, right? So you have bad shot noise at low expressed genes, so they're a problem. Okay, you have technical noise from sample preparation and sequencing. Maybe we can ignore that if someone is good at generating RNA-seq data. But then you have biological noise, the biological differences between the samples, and that's what dominates for the highly expressed genes. Okay. So you've already seen the negative binomial. So basically, you know, there's this combinatorial definition, but that's not the one we think about. We think about the negative binomial as an overdispersed Poisson, where the variance is the mean plus this dispersion coefficient times the mean squared. Okay, so, and this is the overdispersed part. Yeah? Just some examples for, so you have two parameters instead of just one, right? And that allows you to fit your data better and fit the fact that you are more dispersed than Poisson. Okay. So this is pulled out of DE-seq. Okay. Again, every dot here is a counting bin where you have the mean for the bin across the replicates. And then you have a procedure that, based on just the replicate counts for that counting bin, is estimating the dispersion, and it's called it's a procedure called the Cox-Reed dispersion estimate. So apparently a bit complicated, so I don't get into that. But what you see, right, is sort of this: your dispersion is smaller as your mean increases, but it's still there, right? It doesn't go to zero; it's still there, and whatever you do, if you fit this line, you'll have ones that are over-dispersed or under-dispersed relative to the line. And what DE-seq does is it fits this functional form, right? But it's always bumping up. So if you're down here, then it'll bump it up to the line. And if you're up here, it leaves it alone for doing statistical tests. So it's always the higher dispersion estimate that it uses when it's doing statistical tests. Okay, for modeling the read counts. Yeah? Okay. Right. Okay. So before we get to GLMs, let's just think of the simple, you just have one factor, okay? So you have, you're looking at a gene or a counting bin, ClipSeq events, and you're looking at the read counts between two conditions, condition 0 and condition 1, okay? And you're assuming that in both cases, you have a negative binomial, you have a scaling factor times the mean, okay? You have the dispersion sort of looks like this, right? This over dispersion with this alpha. And what we're really doing is we're saying in condition 0, we have a certain log mean. We're going to estimate this beta. In condition 1, we have beta plus this other factor, which we think of as like a log fold change between condition 0 and condition 1. And we're just asking, is this 0? Yeah? So the formulation, we have some interceptor baseline log expression, right? And we're asking, do we need another term, this baseline plus some, do we need this weight to be non-zero to explain the difference between condition 0 and condition 1? Okay? The null hypothesis is that it's 0. The means are equal, right? The log means are equal. And what you do is it's basically a likelihood ratio test for, you have a bigger model where this isn't 0, you have a smaller model where it is 0, and which one has higher likelihood, and can we reject the null hypothesis that this is 0? Yeah? Okay. So, yeah, and then you do that test and you can see, you can color in where the differentially expressed genes are. You have large dispersion and large, your variance is large because relative to the mean at low mean. So you need to see really big log fold changes to see differential expression, and you have more sensitivity at higher mean. So it's not, it's not so different maybe than the pictures you've seen of microarray data, but the test is, the underlying test, the statistics are different, the model is different. Okay, so finished. So the summary, you kind of have these two noise ranges. So you're modeling it as an overdispersed Poisson, but you think of the shot noise dominating at low expression and the biological noise dominating at high expression. Yeah, what to do about biological noise? You need more biological replicants, right? So, deeper coverage of 3 samples isn't going to help you, but okay. All right. GLMs, let me, I think it's getting a bit late, but let me just give you the idea and some concrete examples that come from the ClipSeq and APA. All right, so if you've used the Lima package, I don't know, has anyone done, yeah. So, they have all these linear models. So you after you log transform array intensities, you assume they're normally distributed, and you use old-fashioned linear models. Okay, so we can't transform our data and make it normally distributed with read count data. So we need an appropriate generalized linear model. Okay. So like Lima, you're fitting a GLM for every gene or event, but you share information about the variance, about the dispersion estimate. Okay, so a similar idea. Yeah, okay, so if you have a linear model, okay, you say that you see all these observations like gene expression, right, in different samples, 1 through n, and you say each of them are a random variable normally distributed, but the means there's a systematic part of the noise that depends on factors like was I wild type or mu155 knockout? Okay, so when I'm writing these, I'm going to just think about I'm going to think of these as factors. They're going to be 0 or 1. Is the factor true or not? Okay, and I think of the mean as a linear combination of these factors, and then the noise part is normal. Okay, so we have to change some things to apply this idea of a systematic part and a noise part. The noise model is different. The systematic part is kind of the same. We don't have a linear relationship between the mean and, the mean isn't a linear combination of factors. We're going to need a link function. For us, it's going to be log. So the log mean is going to be a linear combination of factors. Okay? So here's an example, right? So I think of having a design matrix, right? This is describing, in terms of the factors, all my experiments. So I have an experiment, so this is just my intercept, so that's my beta 0. Did I use linker 1 or linker 2? So in some experiments I used linker 1, in some experiments I used linker 2. Was it wild type or was it knockout? Sometimes it was wild type, sometimes it was knockout. Okay? That's my design matrix. So those are my factors. Yeah? And then what I'm doing is I'm expressing the log mean. So for this event, linker 2, knockout, it will look like my intercept, I'm linker 2, so this factor is in. I'm knockout, so this factor is in. Okay? If I were wild type, I would not have this factor. If I were linker 1, I wouldn't have this factor. I would have a 0. Does everyone see how it works? Okay. So we can't use the linear model. We use the generalized linear model. Do I have some? Okay, so let me skip this. Okay. Yeah, so the basic idea is for a wide range of distributions, you can use generalized linear models. You need the appropriate link function. So it's not that your mean is directly related to the factors. You need this link function, right? So we say, okay, the noise part is negative binomial. The systematic part, okay, so I think I don't actually want this here because I've put the scaling factor here. I just want this to be equal to this. EdgeR and DC kind of do it slightly differently, but you have your log means are a linear combination of the factors, and you have this scale. There's an offset that is your scaling factor due to library size. But I can put it, I think with this convention, I shouldn't put this here. Okay, and then you're fitting, just like the F-tests kind of in linear models, you have the smaller model. So if I take into account the linear effect, I have one model, or so the linker effect, I have one model. If I also have the wild type versus knockout, I have a bigger model. Do I need the bigger model? Okay, so so the null hypothesis is that there's no differential binding, that this is 0. The smaller model is just the linker effect. The bigger model is that there's an effect between wild type and knockout. And I have a likelihood ratio test. I want to know if I should reject the null model, if I need this to be non-zero. Okay, so all the tests have that kind of flavor. Yeah? Okay, so last one. Okay, last one, back to this, maybe this will make more sense. Okay, so every event, we have the read counts that we observe in the replicates, okay? We're estimating this log mean, and we're saying that the systematic part, the log mean that we see for an event is a function of gene expression, so this factor, just the gene factor, the event factor, you know, so it's, a condition-specific expression change, and this interaction term. Okay, so we think of it intuitively as, you know, I see this log mean, I get a certain kind of baseline expression, I get a certain mean usage, log expected fraction of the reads that we have here, I get a log expression change, and then I want to know, do I need another factor? Yeah?
I'm not trying to understand why you G and P, it seems to me like it's the same thing, the first two factors.
So this G is for, you think of it just as an intercept, but conceptually we're thinking of, you know, we're only testing this event, one event at a time. So we think of it as we have some baseline gene expression, right? And then, so log expression reads. Okay, so we're fitting these events together. So we have, okay, so the correct thing is all the events for the gene go into this model, but we're only testing whether the interaction term is 0 for one event at a time. Okay, so we need this because this explains the mean not only for this event, but this event, right? This explains the fraction of reads not only for this condition, but also this condition. This tells you the condition-specific scaling of all the events, right? And then we're only testing one, so we choose one event and we test whether this particular gene's interaction, like whether the interaction coefficients are 0. So they're tied, but does that make sense? So we have all these observations, but we're only testing one set of interaction terms. Yeah. Yeah. Okay.
So you're testing a separate model for every gene, right? Because the only way the genes are connected is, I guess, through the estimation of the over
dispersion
parameter. That's the only
yeah.
way to talk to each other. Right?
Yeah.
But then, given that you have that over dispersion parameter fitting this model, you know, the number of data points is as you know, just a little bit larger than the parameters. Right? You have some replicates, right? But
Okay.
it's
So the actual hidden part that made this analysis tricky, and the reason we actually had to hack DXseq, we only have some replicates. We don't have we have replicates of MCF10A and some RSLDine data, right? And we don't have replicates for all our tissues. So what we had to do was assume that we had to get this common dispersion model that was estimated from the replicates we had, and we actually inflated the dispersion. So we were more conservative, and we checked that on the replicates that we had, it was making sense. But then we're making an assertion that the dispersion across tissues is similar to the dispersion across, you know, these experiments that we had replicates for, which, you know, is all we could do, but it's not ideal. Like, ideally, you would have at least 2 replicates.
There's like a
for,
record, right?
Yeah,
Two individual measurements of 2 tissues would be
The absolute lower bound is that you need one condition with 2 replicates, right, for DX-seq to try to do the Cox-Reed estimate. But it's not ideal, you know, like ideally everyone should have multiple replicates for every condition. But this is, yeah, so this
But then
is...
those coefficients are going to be effectively like a log of ratios of powers, right?
So I like to write it out like this because if you look at it, you can think, you know, basically if you're looking at, you know, this term, it's like a log gene expression ratio. So these numbers have some meaning, but they don't have so much meaning. Like it's all relative, okay? So this coefficient is like your intercept is arbitrary. I could shuffle weight from one to others, right? So it's all relative to some reference sample where the gene, you know, you have some reference example where the expression is 0 and it's all relative to that. So these are only defined up to a constant, and you can shuffle weight around.
That's the same in lemma, I guess, right? Or
Yeah. So it's just you know the nature of the math.
Right.
So
My question is something I'm curious about. The over-dispersion, because you have all the parameters specific to one gene, is it fair to say that the over-dispersion doesn't change the estimation of the data, it only changes your p-values?
It changes the p-values, yeah.
Okay.
So the dispersion, you know, your dispersion, it's based on the mean read counts for that event, right? And that's what goes into the tests, right? So, I guess also for the other events that are all modeled at the same time, right? Yeah. So and yeah, that's the only combining. Yeah. Yes.
I have a question that might be misleading. By the fact that you're filtering a lot texts that are not mapping to specific positions, aren't you somehow like compressing the biological variation? Like, or some part of the
Okay.
ways that would be otherwise?
So I don't think so. So I think, you know, so the reviewers asked the same question, you know, like what are the effects of the filtering? And basically the findings are all the same. So you have to filter some, you know, you can't say that one read is an event. It's just not real, right? But whatever filter, if you just filter for 1% usage or 1 TPM or 2 TPM or 3 TPM, these statements about it's always coming in the same place, right, are still true. And if you have these really small events, they will never, if you see 3 TPM, 1 TPM, 0, they just don't have the read coverage to be considered significant difference. So they're all basically 0 or there's no change, right? There's no significant change. So I don't think that even including all these low events, and actually what DX-seq will do is if the event has 0 coverage, it's just excluded from the test. So, you know, you only test in the conditions where you see reads, right? Okay. Any other questions? That's all I was going to say about this stuff.
Well, so you're done.
Yeah, I'm done.